Today's video will be talking about biotechnology. You can understand by the term bio and technology. When these two are combined, we get biotechnology. It is a stream of science that deals with the use of microbes, plant cells, animal cells, or cell components to generate products which are useful to human beings. So here in this biotechnology, we are using microbes and plant cells, etc., using the technology for the benefit of human beings. So it can be for various purposes. So biotechnology is now a very well developed science. The term biotechnology was given by Carl Ehricke. Carl Ehricke gave the term biotechnology in 1917. Now, we have been using, since long back, we have been using products. Man has been using the products, but without realizing the actual method and the function behind it. So, all these activities were basically involving the natural capabilities of microbes and other cells. So, here in this biotechnology, we are utilizing those old traditional methods also, plus incorporating genetic improvement for better production, and other useful products can be obtained which man was earlier not thinking of. So biotechnology has a vast scope, and especially during this time we have seen biotechnology coming up as a very effective branch of science which is helping in dealing with many kinds of microbes and all. Now when we talk of modern biotechnology, modern biotechnology is refers to the production techniques that is based on genetic engineering or recombinant DNA technology. So we have basically biotechnology will be using genetic engineering and the recombinant DNA technology. Now we'll try and understand what is meant by genetic engineering and recombinant DNA technology in the later sections of the videos. But first let us understand the principles of biotechnology. EFB, that is European Federation of Biotechnology. European Federation of Biotechnology, or EFB, has defined biotechnology as the integration of natural science on an organism's cells, parts thereof, and molecular analogues for production and service. The definition this definition which is given by EFB considers the traditional as well as the modern molecular biotechnology. Now when we talk of the modern biotechnology has two main techniques. Now the two main techniques which are involved in the biotechnology is, first is the genetic engineering. So we have genetic engineering as the first. These are the techniques. So we'll first understand what is what, and then we are going to analyze. First is the genetic engineering. That is the first step. And the second is the biochemical engineering. Now what is meant by genetic engineering? This is biochemical engineering. What is meant by genetic engineering? Genetic engineering actually refers to all those techniques which change the chemistry of the genetic material, that is the DNA and RNA. So they are actually those techniques which are bringing about change in the DNA or the RNA. And how we are doing it? By the addition, by deletion, or making certain changes to introduce that into new host organism, and then changing their phenotype. So what are we doing over here? Genetic engineering, as the name suggests, the DNA and RNA are manipulated, we can say. The DNA and RNA get manipulated. These are the genetic materials. So these get manipulated, and this manipulation can be done by either the insertion or deletion or by altering. So we can change to introduce this DNA into the host, and then to change their phenotype. This is what we do in genetic engineering. Now, biochemical engineering is that part of biotechnology. This is again that enables the maintenance of sterile conditions. Sterile means contamination-free zone. So this is these are the techniques which maintain biochemical engineering. This maintains the sterile conditions, and so that the desired microbe or the host only gets multiplied in large quantities, and this is utilized for production of biotechnological products such as antibiotics, vaccines, enzymes, etc. So biochemical engineering is again very, very important, because after the manipulation, this DNA and RNA which has been incorporated into the host is to be multiplied, and that multiplication should take place in sterile conditions, so that no unwanted microbes contaminate the designed microbes, we can say, because we have changed or altered the DNA and RNA as per our requirement. So we can always say that as designed one. So the only we want that desirable host should multiply, and that is achieved through the biochemical engineering. Now when we talk of the principle of genetic engineering, what is the concept behind the genetic engineering? We all know the sexual reproduction in the organisms promote lot of variation, and there is lot of possibility for the coming up of new phenotype. Now these variations are generally useful to the organism, that is individual organism, and in population as a whole. Now the traditional hybridization techniques which have been practiced over the years for the breeding of the plants and animals. Now over there was the disadvantage that unwanted phenotype or undesirable genes also got carried along to the next generation. Apart from the desirable factors, the undesirable genes were also passed on. So what we have to do is, or in the genetic engineering, what do we consider? We remove or we culture only the desired genes, that is not leaving the undesirable ones. So the genetic engineering has come up or has sorted out this problem by the isolation and the introduction of only the desirable genes without the interference of the undesirable genes into the target organism or the host. So what are we doing over here in the genetic engineering? Simply we are using the, like previously also the breeding techniques were used, but in those techniques, along with the desirable one, the undesired traits also passed on. So genetic engineering ensures that that only the desirable genes along with the desirable phenotypic characters should be isolated, allowed to develop in the host, that is target organism, and without the interference or without the transfer of the undesirable genes or undesirable genetic material. Now, this problem, or this is how it is achieved in genetic engineering, since we are talking of only the process today and just trying to understand what is the technique behind the biotechnology. So the problem, or this the solution to obtain the pure genetic material or the desirable genetic material, there are few steps, and the techniques of the genetic engineering include the formation of recombinant DNA, that is rRNA. So what are we doing in the genetic engineering part? We are creating recombinant DNA. So we create a recombinant DNA, that is, we call it as rDNA. That is the first step. So there are certain techniques or methods by using the enzyme. We need to cut that DNA and then have to culture it. Then second is the gene cloning. Then we need to clone that part. So the second step over here is gene cloning. And the third step, which is over here, that is the gene transfer. So these three steps are a part of the genetic engineering. Creating the desirable recombinant DNA, then, or we call it as rRNA, then gene cloning and the gene transfer. Now we have to just get an idea of how the recombinant DNA is created, what is gene cloning and what is gene transfer. Now when we talk, it's a brief introduction which I'm trying to give to you related to the recombinant DNA, gene cloning and gene transfer. Actually it's a complex process and we'll be studying it in subsequent videos. Now over here, when we are talking of recombination or recombinant DNA or rDNA, we know what is over here, the DNA molecule which is made. It is a designed one. Remember, the DNA molecule which is made by combining DNA from organisms of two different species is known as recombinant DNA. So here what we are doing is taking the DNA, desirable DNA from the two hosts, and then trying to make a recombinant DNA. Now the various segments of DNA is isolated, where we use various kinds of enzymes over there, and gene cloning is the process of making multiple copies of the DNA in a host cell. So what are we doing over here in the recombinant DNA? We are first preparing a combination of the DNA, that is rDNA, and then that recombinant DNA is then formulate, that it means multiple copies of the DNA molecule is obtained by introducing that into the host cell. So that thing is there. And the gene transfer, that is the process of introduction of the recombinant DNA into the host cell. Now as we will be progressing with the various processes, we'll be studying about this technology in detail.